Hello everybody, today I'm going to be going through the Enzymes in Action lab with you. If you're at home, that means that you aren't able to participate in this lab, but no worries, we'll make sure that you get to do the lab next time. So we're just going to go through with you. I'm going to read some things. Sometimes I'll say pause here and answer the questions below, but what I really want to do is make sure that you can see what's happening in the lab. Today's lab isn't a traditional lab that you might think about. It's actually using foam pieces to model or act out what happens when we use enzymes. So it should be pretty easy to follow along. If you have any questions, just make sure you reach out to your biology teacher. Alright, so it says introduction. Enzymes are specialized proteins that catalyze or speed up chemical reactions within cells. The substance upon which an enzyme acts is called a substrate. Substrates are small molecules. So enzymes are able to accomplish catalysis without being consumed in the reaction, and it catalyzes a specific chemical reaction. To catalyze means that you kind of jumpstart that reaction or speed it up. Right, so the first thing we're going to take a look at is catabolism. So I'm going to get out those three pieces that they show you here. All right, so I have this gray foam piece, the green foam pieces, and orange foam pieces. It says the gray foam piece is a model of an enzyme. Place it with the A label facing up. Assemble the two green pieces into a single unit to model the substrate. So I'm going to show you here in a second. It looks very similar to the picture. You can see here I have this gray piece, this is my enzyme, and I have this green piece and it is my substrate. So it says draw and label the enzyme and substrate before the enzymatic action. So what I just showed you, I want you to go ahead and draw. You should pause the video now so that you can draw, and if you'd like to go back to see what I showed you, you're welcome to do that. Alright, now that you've drawn and labeled the enzyme and substrate, it says in the first metabolic action, the enzyme will act on the substrate to break it apart. Experiment with the pieces to model how the enzyme and substrate might interact. So our first thing that the enzyme is doing is it's going to break apart our substrate. Remember, the substrate is the green thing. So you'll see I have my enzyme here and I have my substrate is this green, and it's going to fit right in to that spot. Okay, and it wants to break it apart. So what may happen here is my enzyme will hang on to this little piece of the substrate, and this one will be able to come out, and then eventually this one will come out. So now I have two green pieces. Number 4 says the substance an enzyme acts upon is referred to as the, and you should know this one, but take a second to fill it in, it is the substrate. It says place an X on the drawing of the enzyme and substrate you created on page 1 to show where the substrate binds to the enzyme. So you want to put an X on the drawing that you drew above where that green substrate goes into the gray enzyme. The part of the enzyme that binds the substrate to be acted on is referred to as the active site. Once the substrate is locked into the enzyme, the two green substrate pieces may be easily pulled apart, like I demonstrated. This type of metabolic process is called catabolism, the breaking down of complex molecules into simpler molecules. So my green substrate was my complex molecule, and I broke it into two smaller or simpler parts. That's called a catabolic reaction, when you're breaking things apart. I like to remember that because cats break things, and it's a catabolic reaction. Hopefully that helps. So number 6 wants you to draw and label the enzyme, products, and active site after the enzymatic action. So you're going to draw what I had at the very end of the last video I showed you, when the green substrate had broken apart and become two separate products and left the active site. Make sure you label your picture and pause the video now to draw. Now take a second and pause the video and answer questions 7 and 8. For questions 7 and 8, you should have mentioned that the enzyme didn't really change during this reaction. It's still a whole gray piece, so nothing happened to it. And you might have mentioned that it's advantageous for the enzyme to remain unchanged because now we can use that gray enzyme all over again. So it can be repeatedly used. It says, note, a real-life example of catabolism occurs when the enzyme sucrase breaks down the substance sucrose into glucose and fructose, which are monosaccharides. Something to note is that enzymes almost always end in ASE, or ACE. And usually their names are describing what they break apart or what they interact with. So sucrase breaks down sucrose into two smaller pieces. It says, given what you know about catabolism, identify the following components in the model illustrated below. So you need to label each of these: enzyme, substrate, products, and active site. Pause the video now and label those. You should have labeled the following things. You should have labeled A as the enzyme. You should have labeled B as the substrate. You should have labeled C as the products and D as the active site. Notice that D is where the substrate binds to the enzyme. All right, our next section is about the induced fit model of enzyme action. In 1958, scientist Daniel Koshland Jr., PhD, proposed the induced fit model to describe enzyme-substrate interaction. This model suggests that enzymes are flexible structures in which the binding of the substrate results in small changes to the shape of the active site, maximizing its interaction with the substrate. That was a lot of words. Let's break down what that means. Enzymes are flexible, meaning they're bendy, right? They can kind of change a little bit. When the substrate binds, it kind of changes the shape of the active site so that it can really hug it. I'll show you in a second what actually that means. Okay, so you can see here let me put these guys back together. Boop. Okay, so I want you to watch really carefully, especially like right here, as I push the substrate in. Let's see if I can get a good angle. Do you see how it kind of is bending and then poop, it pops right back in place? That's our induced fit model. The idea is that the enzyme kind of had to bend open a little bit so that the substrate could fit in there, and then once it was all the way in, it kind of hugged it back together so that it can touch all of the substrate. Take a second now and answer questions 10 and 11. All right, moving along. Anabolism, or anabolism. You can say it however you want. Tomato, tomato. Enzymes may also bring substrates together to form a final product. This metabolic process is called anabolism, the building of complex molecules from simpler molecules. So this is when you take two smaller pieces and put them together and make a big piece. Use the gray foam piece and the orange foam pieces, I'll show you those in a second, to simulate an anabolic process. So anabolic is when we're building. The orange pieces should not be assembled prior to anabolism action. All right, so let me show you what we're looking at. Okay, so I have my enzyme, still using the same enzyme here. Get that green away, I don't need it. And I have these two substrates, so I have two substrates. Okay, and so what's going to happen in this reaction is I'm going to have this substrate first enter my enzyme, and then this substrate, yeah, that's right, enter. And then after the reaction has occurred, they are going to leave together as one final, bigger, more complex product. Okay, so number 13 wants you to sketch and label the enzyme and substrate prior to enzyme action. So that's at the very beginning of what I just showed you. So take a second, pause, and draw what I just showed you. Once you've drawn for 13, 14 says to place the small pointed orange piece into the enzyme and join the larger piece. I already showed you that. Number 15, in the space below, sketch and label the enzyme and products after the enzyme has acted on the substrate. So the ending of my video, when the two orange substrates are together to make one final product. So go ahead and sketch that now. Enzyme inhibition. Competitive inhibition. Place the gray A, red D, purple F, and blue G foam pieces on your workspace. Got it. Which two pieces may fit into the active site? Well, let me show you. I've got, boom, we know that fits. Red is good. And then purple fits as well. Yeah, it's a little different. We'll talk about that. But it fits in the active site. But my blue does not fit in the active site. That's got a lot going on. It doesn't fit. Can the red D substrate bind to the active site if the purple F piece is bound to the enzyme? Great question. If my purple piece is in there, can my red piece come bind? No, it cannot. Create a sketch using the foam models to illustrate competitive inhibition. Competitive inhibition is when a substance binds in the active site and prohibits or blocks the normal substrate interaction. So you need to create a sketch showing how that purple piece being in the enzyme blocks the red piece. So that competitive inhibitor or purple is blocking my red substrate. Once you've drawn that, predict what might happen in a cell if the concentration of the competitive inhibitors exceeded that of the substrate. So what would happen if you had a ton of purple competitive inhibitors and very few reds? Are you going to be able to have your chemical reaction? Is that red substrate going to be able to be broken apart? Enzyme inhibition. Non-competitive inhibition. A non-competitive inhibitor impedes enzymatic action by binding to another part of the enzyme. So it stops the enzyme from working, but it does it by binding to a different part of the enzyme. This second site, known as the allosteric site, is the place on an enzyme where a molecule that is not a substrate may bind, thus changing the shape of the enzyme and influencing its ability to be active. Okay. So what I want you to do first is predict where the blue G piece may bind to the enzyme non-competitively. Once you've done that, go ahead and take a look. I'm going to show you what happens. Get rid of that. Okay, so this blue piece is my non-competitive inhibitor, and what it does is it binds over here in this little funky place in what we call the allosteric site. So my blue non-competitive inhibitor is going to pop in there. See how nice it fits? But when it does this, do you see it bends our enzyme and changes the shape of the active site? So now my substrate, even if it could almost get in, it actually can't fit there. So my substrate is not able to actually get into the active site. So again, this is my allosteric site. This blue thing is my non-competitive inhibitor. It is blocking my substrate from getting to the active site. Remember, this is called a competitive inhibitor, so it actually takes up the space of the active site so it can't get in there. And then the blue one is a non-competitive inhibitor. The difference in name is based on, the blue is not actually taking up your active site, but it's, kind of going around the outside to bend your shape there, so you're not going to be able to access it anyway. Number 6, take a second and pause and sketch what happens to the shape of the enzyme when the blue piece is bound to the allosteric site. So make sure you draw how it kind of bent up a little bit and blocked my active site. 7, how does this affect the binding of the substrate? Can my red thing bind there? Explain so here at number 7 by pausing. Post-lab questions, predict what might happen if enzyme activity were not regulated within a cell's metabolic pathways. So if it wasn't regulated, what would happen? 2, contrast the action of a competitive inhibitor with that of a non-competitive inhibitor. Remember, that was my purple and my blue. So what does purple do? What's my competitive inhibitor do? And how does that block a reaction? And how does my blue non-competitive inhibitor block a reaction? you might look. Yeah, go for it. Yeah, go for it. You can take a look. I haven't had any classes until these guys. All right, I am back. I'm sorry. Okay, ethanol is metabolized in the body into acetaldehyde. Normally, acetaldehyde does not accumulate in the body because aldehyde dehydrogenase rapidly oxidizes the acetaldehyde into acetic acid. The drug disulfiram inhibits aldehyde dehydrogenase, which causes an accumulation of acetaldehyde in the body with the subsequent unpleasant side effects of nausea and vomiting. Disulfiram is sometimes used to treat patients with a drinking habit. Okay, so it wants you to sketch the action of the competitive inhibitor disulfiram. Be sure to label your diagram with the following terms. Okay, so I want you to try this on your own, and then if you get lost, I want you to come back to this video. I'm going to take a second and make sure I remember this from last year. Ethanol is okay, so if you were lost, you have acetaldehyde. That is your substrate. Okay, aldehyde dehydrogenase. Remember ACE. That's an enzyme. So that's your enzyme. Okay, and that aldehyde dehydrogenase breaks down acetaldehyde. Takes that substrate and it breaks it down into products, which are your acetic acid. Now you have this drug disulfiram, which inhibits aldehyde dehydrogenase. So if you are inhibiting that, it's going to cause the acetaldehyde to not bind with your enzyme, and then it's just going to build up in your body instead of turning into acetic acid. So if you get too much acetaldehyde, then eventually you're going to throw up. So that's why disulfiram is sometimes used to treat patients with a drinking habit. So again, you have acetaldehyde as your substrate, acetic acid as your product, and that's broken apart and becomes your product by using the enzyme aldehyde dehydrogenase. And then you have disulfiram, which is your competitive inhibitor. If you have disulfiram in your system, it's going to block that acetaldehyde from getting broken apart. Yeah, there you go. Okay. How is Kochlin's theory of induced fit supported by non-competitive inhibition of enzymes? Well, if you remember, when I inserted the blue non-competitive inhibitor, it changed the shape of my enzyme. So take a second and write about how this supports Kochlin's theory of induced fit, which basically says that enzymes are flexible. Once you have answer number 4, go to number 5. It says, examine this model of the enzyme shown below. Design a competitive and non-competitive inhibitor for this enzyme. So make sure that your competitive inhibitor can fit into the active site, and then make sure your non-competitive fits into the allosteric site. The allosteric site is the one that looks like a little pentagon. All right, once you've completed that, you are done with the Enzymes in Action lab. If you have any questions, be sure to ask your biology teacher. Have a great day.